Hello everyone, we'll start with the developmental biology. We'll deal with the basic concept so that we are familiarized with basic terminology. So it will be easier in later stages of learning the developmental biology unit. These are topics to be covered. We'll deal with each topic one by one. So basic rule for this topic is topics are there in that unit. They cover or they approximately 25 to 35 marks. Questions are either direct or experimental one. They are based on the direct concept. So our motive is to gain marks, able to get 32, 33, 34 marks or full marks if we are learning it more specific and selective way. So as the developmental biology topic is concerned or this unit is concerned, it is very interesting topic. In this topic, we need not to go in very deep. We have to be very specific and selective in order to secure good marks by this unit. So first term in potency. Potency is what? Potency is basically a cell's ability to differentiate into other cell types. For example, I will show you this diagram. Once the sperm fertilizes that oocyte, now the process of zygote formation starts. After fertilization, embryo formation and zygote formation occurs. These are a step-by-step process. There are various stages in that. So at the morula stage, till morula stage, the cells are totipotent. Totipotent means they are capable of giving all types of cells. Okay, so these are like stem cell, they are totipotent. But at blastocyst stage, once the pluripotent cell, means that inner cell mass, if you extract from the blastula stage, that inner cell mass is basically pluripotent. Pluripotent means that can give many number of cells but not all. This is the basic difference between the totipotent and the pluripotent cells. After the differentiation of pluripotent cell, it gives to other systems which like function to make like proper body functioning, the circulatory system, nervous system, immune system. So these are given by the pluripotent cell. Now, if we talk about the individual system, circulatory system, immune system, nervous system, these are what? Unipotent. They can give now only single type of cell. They can differentiate into single type of cell, that is unipotent. Next term is commitment. As the name suggests, commitment means what? They are committed to form a particular cell fate, a particular certain fate. Means particular cell is committed to form a particular type of cell. In normal conditions, cell fate is there. Means in normal condition, a cell is specialized to give a particular type of cell. If that condition is changed, now the cell is kept in an abnormal condition, then there are two conditions. First is, it can form that original, whatever cell it was forming, or whatever original fate it was, it will be there. But there is also a possibility that it can change, as now the condition has been changed. Now the condition is not the same as it was earlier. So what can be the reasons of cell commitment? There are two reasons basically. First is cytoplasmic determinants. Means every cell, in every cell there is a cytoplasm. So in that cytoplasm, that is basically controlling or governing that cell fate. Second thing is that inductive signal, means a particular cell gets a particular signal to induce something or to respond to a particular signal. So that is the inductive signal. Here this is like a flow chart. Naive cell is there. Now once the cytoplasm determinants and inductive signal, they form like they convert the naive cell to the specified one. Now the specified one get converted to determined. Specified cell are labile, means they can go back, they can revert to that original stage. But once they are converted to the determined one, they cannot revert its fate. That is the basic difference between specified and determined. Once it is determined, it will get differentiated. Stages of commitment. As I told, specification and determination are two stages. In a specification, cell can revert, means it is specified to form particular type of cell. But it is not fixed till now. It is not determined till now. It can revert back. Changes in gene expression are labile and changeable. For example, we take an example like a particular cell has to give a particular type of cell in its natural environment. If I change this natural or culture medium to a non-neutral environment, means a different place, if I keep that particular cell to a different place in the embryo, now as the environment or location of that particular cell has changed, so in that case its original fate will also change. Okay, that is what specification. Determination is what cells are towards differentiated. Now cells are going to be differentiation. Okay, so if we place these type of cell which are determined one, it will still develop as it was in original location. Commitment is fixed. It is not going to revert from its fate. So there is a question which came in CSIR. I don't remember the year, but this is question of CSIR net. So here it is asking for the correct order of early developmental events indicated below. So as I told, first option is what? Determination, specification, differentiation. In the previous slide, I showed you like first thing will be specification, then determination and then differentiation. So what will be the correct answer? C option is correct. First the cells will be specified, then determined and then differentiated. Next topic is induction. So induction in embryology is a process by which presence of one tissue influences the developmental or neighboring cells. Okay, so it is a cascade. Basically, it is a step-by-step process in which a particular cell or a signal influences the development of neighboring or another cell or a tissue. That signal which is generating such effect is inductive signal. There are two, like for this process, for the process of induction, there should be two components. First is inducer and second is responder. Inducer, here as we can see in this diagram, there is an inducer signal which is releasing some inductive signals that are yellow marked. These yellow marked are inductive signals, and these inductive signals are reaching to a responder cell where there are receptors. It is binding over there and showing a particular response. So there should be an inducer cell and a responder which will respond to that particular inductive signal. Types of inductive interaction. So inductive interaction is basically depending on what? First is instruction, whatever the inducer cell is giving the instruction to that responder cell. Second is that environment. As I told in the previous slides, there was example, if we are changing the location, what will be the consequences or what will be the result. So instructive interaction is that it needs a particular signal, means responder cell needs a inducer or inductive signal. That is instructive. It should be instructions. Permissive means area surrounding that particular cell, the environment that is present outside that cell. So it needs only environment to allow the expression of trait. It does not need what? Inductive signal. It contains all the potential. It doesn't need any signal, but it needs a particular environment to allow the expression of that particular trait. Next term is competence. Competence is what? The ability of group of cells to respond to the inductive signal. Means whatever signal is going to be there. For example, like just take an example. There is an inductive signal, but the responder cell does not have a particular type of receptor that can bind with that inductive signal or that induces. So what happens? That cell will not respond to that particular signal, whatever coming from the inductive signal, right? So the ability of that cell to respond to that inducing signal is known as competence. Competent cell has what? Receptors, all necessary secondary messengers, the environment to respond to the inductive signal. This is a question which we'll also read in, like once we'll read the lens formation, but here I have just because these terms I am telling you. Permissive I have already told you. Instructive interaction I have already told. Epithelial mesenchymal interaction is just a type of interaction. Induction and competence I have told. So now we can take this question. So this is a question which was asked in CSIR NET December 2014, where they were like, this question is what? Lens formation requires sequential event whereby the anterior neural plate signals the anterior ectoderm to promote secretion of taxics, which render the anterior ectoderm more receptive to secretions from the optic vessels. The above can be best explained by the following phenomenon. Okay. So here, as we can understand from this question is what? There is an induction. Means there is a anterior plate is there that is signaling some anterior ectoderm to promote the secretion of taxis. So there is an induction. Means there is an inducer that is inducing. Instruction is there, no doubt, but only instruction is not there. So first option is what? Asking instructive interaction only. So no, instructive interaction is not there. Only there is also an inductive signal, means there is an inducer and there is a responder also which is responding to that, and that is the property of that responder is what? Competence. So for the answer, as far as the answer is concerned for this question, so D option will be correct. So what it is saying? Induction and competence. Means there should be inductive signal, that is inductive signal is what? Anterior neural plate is signaling some anterior ectoderm to promote the secretion of taxics. That is the inductive signal is giving by that. Means induction is occurring over there, and then anterior ectoderm more receptive to the secretion from the optic vessel. It should be receptive. So it is competent enough for that. So this is how the lens formation. The lens formation will deal with the when we read the lens formation later slides. Next term is differentiation. Differentiation means it is a normal process by which a less specialized cell undergo maturation to become more distinct in the form and function. That is called cell differentiation. As I told in the first slide where I was telling what a unipotent, pluripotent, particular type of cell. So differentiation is a process in which a cell changes to another cell type. For example of that is what? Development of a single cell zygote to the multicellular embryo that further develops into a more complex multisystem. So there is a stem cell. Stem cell is what? At the morula stage, the cell are totipotent, means stem cell is there. Then further after differentiation, they form sex cells, muscle cell, fat cell, bone cell, blood cell, nervous cell, epithelial cell, immune cell. These are all what kind of type? Unipotent cells. So this is what? Differentiation. That's all for today's topic. Thank you. So first term is morphogenesis. Morphogenesis is what? Morphogenesis is a biological process that causes a cell, a tissue or an organism to develop its shape. For example, particularly after fertilization, means a sperm fertilize the oocyte. Now the whole steps or whole cascade of events that are causing to form an embryo to develop its shape, means the last form is what? Embryo. So whatever the cascade of events or biological processes that is letting to develop its final shape is morphogenesis. Morphogenesis also describes the development of unicellular life forms that do not have an embryonic stage in their life cycle. Means it not only deals with the multicellular life forms where there is a very steps and there is embryonic stage, then there other stages are involved to form a particular last fate or last shape of that particular cell. It also describes the development of unicellular life forms. Means single cell is developed. So it also covered that also inside morphogenesis. Morphogenesis is what? Chemical agent that is able to cause or determine morphogenesis. Any agent or any chemical that is causing morphogenesis is what? Morphogenesis. So for every process in developmental, either it is lens formation, either it is a vulva formation in C. elegans, or any other process that we are going to study under this unit, so there will be particularly a chemical agent or a morphogen that is creating, that is causing the morphogenesis or determining the morphogenesis. That agent is what? Morphogenesis. Type of specification. In previous class, I told what is specification. Specification is what? Specification means a cell is Cell's fate is not fixed. It is not fixed yet. It is very labile and changeable. It can revert its fate. That is what is known as specification. So there are three types of specification. First is autonomous specification. Second is conditional specification. Third is what? Essential specification. So I will deal with each and every with example in later slides. First is what? Autonomous specification. Autonomous, as the name suggests, it is automatic, it is autonomous, it is depending on its own, it is not depending on any other environment or in any signal. So autonomous specification is what? Here cell fate is determined before fertilization. Means morphogenetic determinants, whatever the morphogenetic determinants that are present in ovum, that is determining the cell fate. That is what? Autonomous specification. That is controlling by self. Morphogenetic determinants can be what? Whatever the mRNA, proteins that cause cellular commitment that is present in that cytoplasm will govern, will control the cell fate. So these will be morphogenetic determinants. Then this type of development is called mosaic development. Mosaic means what? It is independent. It is independent of self-differentiating parts. It is also it is independent even to its self-differentiating parts. Means a particular cell will be differentiated, particular cell type will be differentiated into other cell types or other cells. So it is even independent of that also. It is mosaic, means it cannot revert its fate. It will form a particular type of cell which was forming earlier. Here, animal pole is there. There is a different types of cell that are forming. Means it is showing the position. A4.2 is what is forming ectoderm. B4.1, that is posterior part, it is forming mesenchyme and muscle. So here what it does, ectoderm cell is there. They divided that A4 and B4.2. So now also this A4.2 and B4.2 still will form ectoderm separately also. Their fate is determined. It will not change. If you separate these from each other, then also it will form the ectoderm. The same goes with that A4.1 and B4.1. They will still form the endoderm. If you separate also, then also it will form endoderm. It will not form the. So it will form the endodermal cell. This is what? Autonomous specification. Second type of specification is conditional specification. Conditional specification is, here cell fate is determined by the conditions surrounding the cell. Whatever the surroundings, environment is present outside the particular cell, that is controlling the cell fate. So morphogenetic determinants in that case will be inductive signal. It will be signaling among cells within the embryo. This type of development is not mosaic development. This is regulated development because cells of an embryo can change fate based on the conditions within an embryo. Means whatever the condition that is present inside the embryo, that is controlling the fate, that can change the fate. Cells that can change their fate. It is conditional. It is dependent on particular conditions. Means if this condition is there, then it will form a particular type of cell. If this condition is not there, then it will not form that particular type of cell. Here it is an example of development in frog. So here I am just giving the brief. We'll read it very in detail when we read the frog developmental part. So here it is happening what? Let's see. First thing is that it is a conditional specification. Why? What a cell becomes depends on its position in the embryo. Means its fate is determined by the interaction with the neighboring cells. If cells are removed from the embryo, the remaining cells can regulate and compensate for the missing part. Here, first example is what? Here is a blastula. Normal back cell is there. It is denoted by the red mark. If you transplant this normal back cell to the belly region, now this red part will form where? In the belly region. Okay. And this earlier what it was forming, the normal back cell was forming in the back. on the back. When there was no transplantation, it was forming a normal development. So here it is the red part. Here we can see the red part is where on the back. But when we transplant the back cells to the belly region, now the cell will be there where? At the belly region. So what a cell will form? It depends on where? Position in the environment, where it is present. In first case, it is presenting in the back cell, it is forming in the back. In other example, other diagram, it is where it is transplanted to a belly region, now it will be formed in the belly region. In second example, in part B, here we can see what? If you remove a particular type of cell, means if you remove some of the cells or remaining cells, if you remove the particular small part of a cell, then all the other remaining cells will compensate with the missing part and will still form the normal development. It will not change to the abnormal development, still the normal development will occur in spite of being removing some cells part. Okay, so this is conditional specification. Next is syncytial specification. Syncytial is what? Here, this is the formation of head and tail. So this will also read in very detail how the head and the tail are forming, what are the morphogens that are controlling the head and tail formation. But here, for example, I am telling syncytial specification, it is basically a hybrid of autonomous and conditional specification. And here the example will be basically from the insects. This method involves the action of morphogen gradients. There are no cell boundaries in the syncytial. These nuclei works in concentration dependent manner. Okay, here as we can see an example, bicoid nanos, these are all morphogens. Bicoid controls the head formation, means the anterior part. Nanos control the posterior parts, means the tail part. So here whatever the concentration is there, that is controlling, means concentration of morphogen is controlling basically the development. In fertilization, bicoid is there. Bicoid is forming what? As I told, head. Nanos is forming what? Tail. There is a gradient, means wherever the concentration of bicoid is there, then it will form anterior part or head. Wherever the nanos concentration is higher, it will form the posterior part, tail formation will be there. Okay, acron, that is head, then head forming region, then there will be thorax, abdomen, then abdomen, and then tail part. But bicoid and nanos concentration, these proteins concentration are determining what will be the anterior part and what will be the posterior part. So this is basically, there is no boundary of the cell. Okay, the nuclei works in a concentration, means wherever the particular concentration of a particular protein is higher, they will form a particular type or particular part of the cell. Another type of protein is higher in particular area, then it will form that type of part of that. Then cell fate and cell lineages. Cell lineage is what? It is describes the developmental history of a cell from its birth under unit until its final division and differentiation into particular cell type, which is known as cell fate. Means from very initial, from a dot to form the final cell structure, final cell shape, that is cell fate. Cell fate is basically determined by the actions of numerous cell intrinsic as well as extrinsic factors. Means an intrinsic is what? Inside the cell. Whatever the condition is present inside the cell, that is controlling it, as well as neighboring environment is also controlling, that is extrinsic factors. So these are the examples. There is an adult stem cell. They are differentiating in hematopoietic stem cell, epithelial stem cell, neural and mesenchymal stem cell. Then mesenchymal cell are further differentiated into adipocyte, chondrocyte, osteocyte, fibroblast, mycocytes. Then adipocyte is further forming what? Fat tissue. Chondrocyte is forming what? Cartilage. Osteocyte is forming what? Bone. Fibroblast is forming what? Tendon, ligament. So these are the final, from the adult stem cell to the muscle formation, there is a stages. Okay, So these is cell fate of muscle formation will be, if you call about the cell lineages, then you have to tell like adult stem cell differentiated into the mesenchymal stem cell differentiated into the myocyte, then myocyte formed after differentiation, that is muscle cell. So cell lineage is what? Zygote means first fertilization occurred, zygote formed, then during the embryo formation blastocyst is there, they differentiated to the endoderm, ectoderm, mesoderm, then mesodermal part of the like in the whole cell, in the whole body. Mesodermal part is what? Heart, muscle, kidney. Ectoderm is what? Skin, neuron. Endoderm, lung, intestine, liver. So whatever the final fate, final shape of the cell, it has passed from the particular cascade of events. These are called cell lineages. What are the result of particular differentiation? Stem cell, as I told, at the stage of morula, they are very totipotent. They can differentiate into all type of cell. So stem cell are basically what? Body raw material, cell from which all other cells with specialized functions are generated. Here, as I told, from the blastocyst, that means from the morula stage, the inner cell mass stem cell that is differentiated into a type of cell, that is the cytoplasmic determinants. Cytoplasmic determinants, the molecule that are localized in the specific cytoplasmic regions of the unfertilized egg or zygote and affect the cell fate decision by segregating into different embryonic cell and controlling distinct gene activity in these cells. Means cytoplasmic determinants are all those molecules that are present inside the cytoplasm of that zygote or any cell that is deciding the cell fate are known as cytoplasmic determinants. In particular, egg, if we talk about the egg, such determinants are what? Maternal mRNAs and proteins. In dividing cell, this lead to the asymmetric cell division is there, daughter cells form, then they differentiate to the particular different cell type that are cytoplasmic determinants. They are basically localized to a particular place, particular like part of that cell. Here, this cytoplasmic determinants, as I told, there is a sperm and then there is a egg. They are fertilizing, then they are forming a zygote. Mitotic division is occurring over there. Then they are forming two-celled embryos. Thank you for watching.